So before, I know it sounds like a fancy name. So before we go any further, I'll do some explanation. This paper is basically, it's very interesting article by a Chinese group. And it is in essence about the potential factors involved in periodontal disease and tissue destruction, which is relevant to all of us, obviously, but could also be relevant to implantology. The premise is that periodontal disease is a situation where you have an overactive and uncontrolled immune response in patients who are susceptible to it. And they also bring in type 2 diabetes mellitus, because this increases the incidence and exacerbates periodontal disease, potentially through its hyperglycemic and dyslipidemia. So that's the premise they work on. But they also bring in the macrophages. One of the first inflammatory, one of the first lines of defense in periodontal disease is through macrophages, which is why they're bringing this in. And so although it's the first line of defense, macrophages can also have a destructive effect as well. So just before we go further, I just want to do a wee bit of revision on just this. Can you see my PowerPoint now? There's a little slide. Yeah, okay. So in essence, if you, just to remind you, monocytes become macrophages in tissues, and there is a gradient which is widely spoken of, kind of macrophage. M1 macrophages tend to be more pro-inflammatory, and they release kind of pro-inflammatory cytokines, as opposed to the other end of the gradients. They can be polarized towards an M2 macrophage, which is more of an anti-inflammatory macrophage. So just to remind you what the difference between M1 and M2 macrophages are. The other thing before we talk...
Fadi, can I ask a quick question?
Yes.
Do you think they are different macrophages, or are they the same macrophage in a different phase?
I think the latter, from my understanding, is that they can be pushed, for want of a better word, pushed towards one or the other pole, which is why I guess we call them polarized towards M1 and M2, rather than that's it, for its life it becomes M1 and for its life it becomes M2. It's more of a pushing it to one pole or the other. Would you agree with that?
I absolutely agree. I think the original concept of these TH1s, TH2s, the macrophages, the different types of macrophages, was based on the existence of different pools of macrophages. But the truth of it is, I think that monocyte, depending on the environment it encounters, can flex between one stage or the other, depending on the signals it gets. Yeah, I think that's a really important point.
Yeah. Thank you. And actually, that's what is brought up in this paper as well. So the other thing I was going to share before we go further, I'll explain this, because what happens when a macrophage is stimulated? So this is just a little diagram for my thesis, hopefully to try and explain before we go into the paper. If you look at
did you all notice the subtle way he got in about his thesis?
I'm going to milk it every opportunity I get, Prof.
Oh, I feel quite sick. Hands up, all those of you who have a thesis.
Every opportunity. Unashamedly. Thanks for pointing that out.
No, that's fine. Right, so carry on with your thesis.
I'll scroll a bit up on that. No, anyway, this diagram basically indicates that in a macrophage, what tends to happen, they can be primed with various foreign bodies, or things like lipopolysaccharides from bacteria, They can even be primed with some cytokines. But in essence, when you've got a macrophage or that meets bacterial component, whether it's an RNA or lipopolysaccharide, it can get primed through what we call the toll-like receptors. Priming means it's now activating the nuclear factor kappa B gene for making some pro-inflammatory cytokines. Okay, so you get external, in essence, get external factors that prime the cell to start transcribing through NFKB, transcribing some cytokines. And these cytokines for macrophages tend to be, I've just mentioned one here, but in the study they'll mention more. So we've got pro-interleukin-1 beta, interleukin-18, and TNF-alpha. These are in the pro state, so they're not active yet. Through inflammatory processes, it could be foreign bodies or it could be other inflammatory processes, another group of proteins called inflammasome, NLRP3 is part of this inflammasome, gets activated. Okay, and this activation results in an enzyme activation called capsase-1, which takes the pro state of your cytokines to the active state of the cytokines. You'll realize what's the point of me mentioning all this. So in essence, in summary, cells get activated through the NFKB to form pro-inflammatory cytokines. The pro-inflammatory cytokines are then activated by capsase-1. Okay, that's just a wee bit of revision on that process before we go into the study. So going back to this study, they are looking at how macrophages are polarized towards M1 macrophages, and what happens with the process called pyroptosis. Pyroptosis, you will have heard of apoptosis. Apoptosis being a programmed cell death. Pyroptosis is a type of programmed cell death in response to inflammation. So in periodontal disease, people have found higher levels of M1 polarization and pyroptosis. What this study looks at, what is the mechanism of this? What's activating and promoting this? And they look at this AZGP1. The AZGP1 is basically a type of glycoprotein. So if I scroll down, the glycoprotein is involved in lipid metabolism, lipolysis, and does different things in different tissues. But they reckon that this glycoprotein might be one of the factors involved in this macrophage polarization and pyroptosis in periodontal disease. What's the point of that? Well, if that's the case, it could potentially be a target for therapy in the future, even if it's local therapy. So what they've done, let's have a look. They took, in essence, human gingival tissues. They had 20 patients who were healthy, 20 patients who had periodontal disease only, and 15 who had periodontal disease and type 2 diabetes mellitus. So they had some human gingival tissues, but they also looked at some animal studies to do some kind of more detailed analysis. I will summarize their results because obviously it's a fantastic paper with loads of results. But in essence, they found that this glycoprotein, AZGP1 is basically the gene that codes for it, is overexpression of this aggravated periodontitis and promoted M1 polarization and pyroptosis. So they found that in human gingival samples, both mRNA protein expression levels of this glycoprotein were significantly increased in the periodontal and periodontal disease with type 2 diabetes. The expression of this in 3 groups demonstrated co-localized. So they found it with macrophages. CD68 is a marker for macrophages. They found the two co-localized in the same kind of regions. So that was one set of results. Another set of results showed that they used in mice. So they did the same kind of experiment in mice, and they found that if you had... Sorry, we've got somebody that needs to be muted.
Sorry, Fadi, you are on mute, but I muted everyone else.
I muted myself. Okay, great. So I'll crack on. So basically, the next set of results they did. Can you hear me now? Yeah.
Yes.
Great. They used knockout mice. In other words, mice that didn't have the gene for this glycoprotein. The AZGP1 gene was removed, so they weren't able to produce this. And they found that this reduced alveolar bone loss and inflammation with periodontal and type 2 diabetes associated periodontitis. So if you don't have the gene, it reduced bone loss and inflammation. They also found that with the lack of the gene, the overexpression of the gene or the knockout significantly promoted or inhibited the activation of that inflammatory gene. If you remember that diagram I showed you earlier, where from my thesis, where the NF-κB gene produces the pro-inflammatory cytokines, right? If this glycoprotein overexpression increased its activation and its knockout significantly inhibited its activation. So we had less pro-inflammatory cytokines. So it has the effect, they checked the effect at genetic level. Next set of results showed that basically not only they saw it from bone loss and tissue destruction, the glycoprotein effect promoted M1 polarization and pyroptosis via activating that inflammasome capsase-1 signaling we talked about. The way they showed that they tried to block the effect of capsase-1, and they found that actually the AZGP1 not only increases the genetic, the transcription of NF-κB, but also the pathway through the inflammasome capsase-1 signaling. So that pathway diagram I showed you seems to be, to a certain extent, controlled by this glycoprotein. Okay, so the point of this, their conclusions are as such. They found that glycoprotein was primarily co-localized with macrophages in periodontal tissues. Its overexpression or its knockouts had the respective effect on periodontal inflammation, bone loss, as well as M1 polarization and pyroptosis. Pyroptosis, basically, as we said, is an inflammatory type of cell death, which releases a lot of material into the tissues that causes more inflammation, is pro-inflammatory as a result of that. They found that blocking the inflammasome capsase-1 pathway reversed the M1 polarization and pyroptosis induced by the glycoprotein overexpression. So indicating again more that this seems to be the aggravation through the glycoprotein seems to be through this capsase-1 pathway. The other thing they found is that the overexpression of the glycoprotein increased inflammation and bone resorption, whereas knockouts reverse effect, underscoring the pro-inflammatory role of this in periodontitis and type 2 diabetes. But they also found it had an effect on the periodontal ligament stem cells as well. It seems to have actually affected the periodontal ligament. Here we go. It had an effect on that. So in essence, there was a correlation with that and the activation of pro-inflammatory cytokines, and this GSDMD is a way that the cell creates pores in the membrane, and it basically bursts, if you like. It swells and bursts and releases a whole set of inflammatory markers. So a lot of this is repeating the same things, increasing pyroptosis. So this is the bit with periodontal ligaments. So glycoprotein type 2 macrophages could also impair the function of human periodontal ligament stem cells, thereby contributing to the progression of periodontal disease. So the conclusion was, the results suggested that glycoprotein, alpha-glycoprotein 1 participates in the pathogenesis of periodontitis and type 2 diabetes mellitus associated periodontitis by aggravating the M1 polarization. So this is the way it does it. It makes the M1 more of an M1 polarization, and pyroptosis. Pyroptosis is the breakdown of the cell, releasing all the inflammatory materials, and that's all done through the capses-1 pathway. The relevance of that is obviously it underscores a possible effect, a possible target rather, for treating periodontitis and type 2 diabetes. There is a huge amount of generalization, obviously, because this alpha-2 glycoprotein is found in many tissues, liver, lung, and so on, and its effects can be different in different tissues. However, this is indicating a potential localized effect that can be targeted. Some of the issues is that they didn't test the polarization, as Prof. you and I were discussing earlier, they didn't test M2 macrophages or start with monocytes to see actually, do they polarize M2 back towards an M1? Do they change the gradient? That would have been interesting to see. And they also could have looked at other inflammatory pathways as well. They focused on the inflammasome pathway. Those were a couple of kind of limitations that they came up with. But otherwise, I just thought it's very interesting because finding out what happens at the molecular level and potential targets could help us with finding potential therapies for that. So I think I've spoken long enough on that, Zoe. I can open it to discussion now.
Fadi, it's Sinjin. Has anyone looked at this glycoprotein? I may have missed this, apologies. Has anyone looked at this glycoprotein in periimplantitis?
Not to my knowledge, and that's the interesting thing.
Is that your next thesis?
Next paper, at least, hopefully we can. That's something worth.
I think that would be worth looking at.
Yeah.
My only worry is, and we've had this discussion before, and I don't know whether colleagues have suffered me saying this previously. Human beings love to simplify what are, let's face it, very complex pathological processes. And in its simplistic form, we always love to know that A converts B, which converts C, which gives D. So if we block B and C, we won't get D. The unknown in all of this is, imagine you did develop some form of inhibitor of AZGP1. Based on the fact that you know it's got a role in periodontitis and type 2 diabetes. The question is, what else would you disrupt? Because of the nature of the human immune system, it's not a linear system, it's a network of redundant activities that get activated. If you block one, another one gets blocked. And that's always the anxiety when you identify one target, is where does that target live within the grand scheme of networks? What other networks come in? Colleagues may remember, some of you who are old enough, maybe you're not, but when I was a young man, there was a drug developed at Charing Cross Hospital, which was supposed to be a miracle cure for rheumatoid arthritis. It was an anti-TNF alpha antibody, and it worked for 6 to 12 months. Patients crippled by rheumatoid arthritis having had this drug, their symptoms lifted to the extent they became relatively free, physically that is. But 6, 9, 12 months in, all the symptoms started to come back again because, of course, other networks that contributed to the inflammatory destruction kicked in and matured. And that's the challenge, isn't it, Fadi, when we have really interesting papers like this, identifying key intermediaries. But the disadvantage of this system is it's in vitro.
Mm.
I'm going to present a paper which is in vitro, and that's a big disadvantage. We really need to know how they operate within three-dimensional physiological systems, which either is an animal, and colleagues will know that animals now are increasingly criticized as non-representative of the human state, or, Fadi, as you and I have discussed, and we're going to have to think of a way to do this quickly, and I've got a meeting this week, it's to create these organoids, these three-dimensional stem cell-based physiological systems where you can see the different cells interacting with each other when you knock one of them out.
Mm.
So I think there's no doubt, as a physiological target, it's really interesting. The question we all have to just bear in mind is, well, what would happen to the rest of the human body if you knocked out this glycoprotein? That would be the problem. I'll shut up now because I can see Hassan. Hassan, are you in Canada?
Indeed, indeed. How are you, sir?
Very well, young man. It's obviously the middle of the night there. I assume it's about 5:30, is it?
In the morning.
Good.
You see, sleep is for the weak. Sleep is for the weak.
Yes,
So,
yes, but they look very strong.
You know what? It's such a beautiful paper, but macrophages is, I don't know, we all share the love of macrophages. As a matter of fact, it's my time only to speak a little bit, but I've just flipped Prof. Crean an email right now to tell him about last week. I was in Nashville in the American Academy of Implant Dentistry presenting about the role of macrophages in alveolar bone healing, right? So, I mean, I was looking into something called FTY720, which is a drug for multiple sclerosis that will trigger up SMP pathways for macrophages to transformation. One thing that when I look at those papers again, is that they did not isolate anything else, right? So they did not look at your interferon presence. They haven't looked at any other interleukins. Is it 4, right? And 6, is it 6? They haven't looked at anything at all apart from this, right? So how can you specifically say that this is the reason for the transformation or the shift to the from M0 to M1? With regards to polarization, my only thing about whether they are the same thing or not is that what they produce. Prof. Crean, what are your thoughts about that? Because, you know, your macrophages too will produce TGF, and they will produce different types of interleukins, right? So how can they be able to do that when they
all carry the same genetic code, and it's which genetic code gets turned on, isn't it? So if you've got a receptor that's pushing you towards M1, then your secondary messengers and your transcription factors, that's the way I think of it, Hassan, will activate the M1 armory. If, on the other hand, the stimulus coming in externally pushes you to M2, the same cell carries the same genetic material. It just turns on a different set of transcription factors and therefore genes. I've become more comfortable with the concept, Hassan, that the macrophage slips from one form to the other. Whereas the impression we all get when we read these papers is that there are different populations of macrophages. Well, at any one time there may be, but they're all basically derived from the mother cells.
Yeah,
that's my instinct, Hassan. I'd love to know if we're wrong.
You think they go? No. I mean, you're definitely not wrong. But you go up from M0, you go from M0 to M1 to M2, but you go back from M2 to M1 again.
Why not? Why not? Because you've got the same genetic code, you're just receiving different signals.
Right.
That's my instinct.
Pages 2, you got a toll-like receptors, you would actually transform into M1 again.
I mean, toll-like receptors are not the only receptors they have, remember that?
Yeah.
So there's a lot of different things that activate them, isn't it? So it's not just the toll-like receptors, there's also endocytosis, isn't there? So they can actually phagocytose things. There are things that can seep in through Trojan horse effects and so on.
It's the whole PAMP and pimp type of relationship going on, you know.
Yeah.
We're learning more and more about how these cells interact with the outside world. And it isn't just, sometimes, as you know, Hassan, it isn't just about the actual chemical composition of the ligand. It may be the spatial arrangement of it. This is a complex world we live in. Yeah. This is a complex world we live in.
Yeah, I think the reason this was interesting, again, it's not because I think we're going to have a miracle cure. I totally agree with what you were saying, Sinjan, is that it's another piece in the puzzle. Just another piece. We're getting, hopefully, a bit closer to understanding that complex network. But
the problem is now, us here today, Hassan is a good example of a thinking clinical scientist, is we're capturing lots. I mean, Hassan presents in a conference about one particular area. You present a paper on this. We capture all this information. What we really need is some genius, autistic data scientist to stand back and somehow get it all together. Maybe even AI. Maybe all this information needs to be pumped through AI.
Yeah.
And let's see what comes out the other end.
It's a whole science field in itself, isn't it? Medical networks, the network science. It's a whole network.
Informatics.